right, so welcome back to Mr. Saucedo's YouTube videos. We've been talking a lot about plants. It seems like we've been talking about genetic material in plants a whole lot. So now we're going to look at human genetic disorders, something that's a little bit more applicable to us. Um, and then if you have any questions, obviously, remember to always let me know. So we have 46 chromosomes. That's 23 pairs of chromosomes. And when I say 23 pairs, a lot of times people think that means that they're identical. They're not identical, right? If we've learned anything from Mendel's work, it's that we do and can have different alleles on different chromosomes, right? And more importantly, though, really only the first 22 are paired like that because the last two are sex chromosomes. So we'll talk about that in a second. So the first 22 pairs, we call those autosomes. And so here are what some of our autosomes look like. And they organized them by height, or by, I guess it wouldn't be height. Well, I guess it would still be height. But how long they are. Maybe length would be the best way of saying it instead. And so you might think, they didn't do a very good job. Yeah, they didn't do a very good job. When we discovered chromosomes, again, this is early in, like we had some powerful enough microscopes to do it, but we did not necessarily have very interesting and nice measuring techniques in order to know exactly the length of each of the chromosomes. So why don't we reorganize them? Because we've done all of this work and everybody knows what chromosome 1, 2, 3, 4, 5. Like we know the chromosomes now. So instead of shuffling them around and being, oh, well, what was chromosome 5 is now chromosome 4, that would be really confusing. So we kept our errors in here. And so this is what our 22 pairs of autosomes look like. They look something like this. The last pair are sex chromosomes. And if you are male presenting, that means that you have an XY. Clearly, these are not the same. So when we say we have 46 chromosomes or 23 pairs of chromosomes, if you are XX, then technically you have something that is closer to, that last pair is pretty much a pair. But for XY, clearly the Y chromosome is very different looking than the X chromosome. Okay. And so in humans, there are two sex chromosomes. We have the X and the Y. Again, in other organisms, they have different chromosomes, and we like to use different letters. Sometimes they have a single chromosome that is a sex determining chromosome. And so again, we have XX and XY. And so those are going to be our primary distinctions, is XX versus XY. That's kind of what we're going to utilize as our terminology. Okay, so autosomal disorders. Those are disorders that are caused by mutations in the first 22 pairs of chromosomes. So there are two types of autosomal disorders. We have autosomal recessive, which is the first one that we're going to talk about. And in an autosomal recessive disorder, homozygous recessive individuals have the disorder. So that means if you have two lowercase letters, you have the disorder. If you're heterozygous, you are a carrier. Now, carriers are different, okay? Carriers don't have any symptoms of the disease. They are absolutely and completely, as far as we're concerned, right, like phenotypically normal, but they could carry a recessive allele, or they do carry a recessive allele that could get passed on to their children, making either more carriers or maybe someone with the disorder, okay? And so an example of that is cystic fibrosis. So that's a disorder that affects the buildup of mucus in the lung, and it is caused by a genetic, I almost said mutation, but I think normally it's caused by a deletion. And so chromosome number 7, there's a gene called the CFRT gene right here. And so just simply not having this amino acid is going to mess up the ion channel in your lung cells' cell membranes. And so because you can't, move chloride ions through the ion channel effectively, you end up building up mucus in your lungs. And so cystic fibrosis is an example of, like we said, an autosomal recessive disorder. And so how does that look for a Punnett square? Well, we have to first again decide what the normal and the recessive letter is going to represent. So we're going to say capital letter C, that's a normal allele, that's dominant. Lowercase letter c, that's going to represent a cystic fibrosis allele. Not having cystic fibrosis, just having a single allele, okay, just having the allele for that. So let's say that both parents are carriers. They're both heterozygous. So remember, we take the letters and we separate them out. And now we have, two capital letters, capital letter, lowercase letter, and then one child that has two lowercase letters. So if we're looking at the genotypes for these, that means 25% are homozygous dominant, meaning that they are 100% normal. 50% are carriers. That means that they are also normal, right? But they hold on to this recessive allele that could get passed on to their children. And then last but not least, we have one child that has cystic fibrosis because they are homozygous recessive. So phenotypically, 75% would be unaffected, 25% would be or have cystic fibrosis. And again, when we say unaffected, we're not separating them out as carriers, because if that were the case, we'd say 25% are unaffected, 50% are carriers, okay, and 25% have cystic fibrosis. So we like to really kind of, for genotype, we take care of the carrier stuff for the genotype, not for the phenotype. Because remember, phenotype is just what trait you present, and so 75% are presenting completely normal. Okay. What is the other type? Well, we have autosomal dominant disorders. And that means that all it takes is a single capital letter to have the disorder. And so homozygous dominant or heterozygous individuals have this disorder. Okay. There are no such things as being carriers of autosomal dominant disorders. Either you have it or you don't. Autosomal dominant disorders, again, there are plenty of them out there. They are much less common, though, than recessive disorders. And actually, why do you think that might be the case? Tell me tomorrow when we actually talk. Okay, good example, Huntington's disease. So Huntington's disease is a disorder that usually leads to a loss of nervous function in adulthood. And this disease normally stays dormant. You can go your entire life without knowing that you have Huntington's disease because it only really affects people when they get into their 40s to 60s. And almost never do symptoms appear before that. And the reason why it is a very interesting disorder is because most of the time, if you're in your 40s or 60s, you most likely have already passed on that gene to your children or onto your grandchildren, and it can cause damage to the brain. Okay. And so Huntington's disease is an interesting example of an autosomal dominant disorder because most autosomal dominant disorders, are pretty serious, but they'll affect you when you're earlier in life and can cause a lot of problems and maybe an early death, whereas Huntington's disease doesn't. Huntington's disease waits until years later when you're in your 40s or 60s before symptoms appear, and at that point, you've probably already passed on that gene to your children. So how do we do a Punnett square for an autosomal dominant disorder? Well, we're going to do this for Huntington's disease. So capital H means you have Huntington's disease. Lowercase h is a normal allele. So let's say that one parent is heterozygous. One parent has one copy of the Huntington's disease causing allele, but that means they have Huntington's disease. Because remember, there are no such things as carriers. And then we have one parent that's totally normal. Okay, great. Well, simply by luck of the draw here, that means that 50% are heterozygous, 50% are homozygous recessive. That means 50% of your children would be unaffected, and then 50% would have Huntington's disease, though. And so you can see why even having a single copy of that dominant allele can have a huge effect on outcome later on. Now, there are other types of genetic disorders out there too, such as the genetic disorders where you have too many or too few chromosomes. So most of these disorders are caused by nondisjunction, and we'll talk about meiosis in a little bit. Okay, we'll talk about it in probably like a week or a couple of days or something. And just like mitosis, where your spindle fibers are separating out your chromosomes. In sperm cell and egg cells, same exact thing is happening. And there are mistakes, there are errors that get made in that process. Most of those disorders are non-disjunction disorders, and that is literally where the spindle fibers make an error. They don't attach or separate chromosomes correctly. And so now you end up with an extra chromosome in one egg cell or one sperm cell, and you end up with one fewer chromosome in the other egg cell or the other sperm cell. And so this is much more common in older individuals' eggs and sperm cells. So when we're talking about, why having children later in life, generally speaking, causes more problems, one of the problems is that non-disjunction is more common. So the likelihood of you having a child that has a non-disjunction disorder increases with age. I think it's once you're 40 or something, or maybe it's even like mid 30s. But here are the common ones that you guys have probably heard of. So Down syndrome is an extra copy of chromosome 21. This is what it looks like if you were to separate out. These are called karyotypes, where we separate out chromosome 1, 2, 3, 4 from a sample of DNA. And you can see here we have 3 copies of chromosome 21 right here. Edwards syndrome is an extra copy of chromosome 18. Again, if we take a look, you can see we have our triplicate here. Patau syndrome is an extra copy of chromosome 13. Right here, unlucky number 13, I guess, right? And then that also happens, though, in our sex chromosomes. So you could have Klinefelter syndrome, where you have 2 copies of an X and 1 of a Y. So you can see right here, you have 2 copies of an X and a Y chromosome. Triple X syndrome, you can see you have no copies of the Y, but 3 copies of the X. Turner syndrome is called XO, where you have a single X chromosome. It is the only genetic disorder where you have a missing chromosome and still can function normally. And then Jacob syndrome is XYY, where you have 2 copies of the Y chromosome. And so that's it. If you have any questions, please let me know.